discovering the intricacies of our world, understanding life. From our smallest plants to our deepest oceans. Observing the breath of life. Reaching people everywhere. Fighting silent battles. And reclaiming strength. For our well-being. So we can keep moving forward and deepen our understanding of biology itself. Krios 5 Cryo-EM, structuring life.
Hello everyone and welcome to today's webinar. I am Rosie Manser and I'll be your moderator for today's event. I really hope you've enjoyed exploring the virtual rooms. You can visit the resource room to find additional information, download, view and read more about the Krios 5 and today's webinar in general. Today we're going to be introducing the latest generation thermoscientific Krios 5 cryo-EM and also talk about advancements in both single particle analysis and cryotomography workflows. We very much encourage you to participate by submitting any questions you may have during the presentation. To do so, you can simply type them in the Ask a Question box and click Send, and we'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical difficulties at any time during this event, you can use the Q&A box and we'll assist you directly. Today's webinar will be in 3 parts. We'll start with an introduction and an overview of the new Krios 5 TEM, and then move to a close-up look at the workflow that runs from the Arctis cryoplasma fib to the Krios 5 TEM. And then we'll have a live Q&A session. So let's kick it off. I'd like to welcome today's first speaker, who is Marc Storms from Thermo Fisher Scientific. Marc holds a PhD in virology and has a long history with electron microscopes, having held several positions at Philips, FEI, and most recently Thermo Fisher. Marc has been involved with the hardware and software development and release of various transmission electron microscopes over the years, as well as instruments for sample preparation. Currently, Marc is responsible for the product marketing of the Krios and some of its key accessories in the context of both single particle analysis and tomography workflow solutions. Marc, we're very much looking forward to your presentation. Over to you.
Thank you very much, Rosie, for the introduction, and welcome everybody for the webinar in which I would like to introduce the new Krios 5 electron microscope to you. However, before I do that, I would like to give a short introduction about how the field has developed, how cryo-electron microscopy has developed, the 2 main workflows that symbolize this: single particle analysis, which has allowed us over the decades, over the years, to solve structures of purified proteins at atomic resolution. In the meantime, having matured so far that it plays an important role in drug development and drug design. The alternative and complementary workflow is tomography, where all the information that single particle actually has provided can be put and can be seen and translated into the cellular context, looking to, let's say, cells, cellular fractions, and tissues. Those 2 workflows have matured quite significantly over the past years, and this slide shows a little bit where we stand with respect to cryotomography in comparison to single particle analysis, which was earlier, let's say, on the rise. We roughly have a 6 years delay, if you like, in terms of the amount of EMDB deposits in the database, which is a typical indication for the maturity and the rise of the technology, if you would like to say. But what is also clear, single particle, of course, still very important in terms of deposits, but tomography is catching up, and especially the last year we see a kind of exponential increase of the number of data that have been published in the EMDB. But there are also other indicators that cryotomography gets more important. You can look to, of course, the leading indicators, the papers in Science, Cell, and Nature, but also looking to the labs that are adopting cryotomography, that have invested into cryo-FEG technology using tomography software. And there we also see very steep increase. More than 50% currently of all the cryo-electron microscopy labs are applying to some extent cryotomography. And of course, both technologies have led to a very wide understanding of the fundamentals of life, if you like, on first of all the visualization of how proteins interact with each other, but also interact with all kinds of other macromolecules, DNA, RNA, lipids. Tomography has given us the possibility to look to cellular components in their native state, being the cell context. And we also see a spin-off with respect to the industrialization of the technology, and that is with respect to the atomic visualization of proteins, which support drug design efforts. And if you currently look to where we stand with respect to development of new drugs and medications and the role of cryo-EM in here, then we can really see that cryo-EM is getting significantly more important. The number of potential drugs that are in the phase 2, phase 3, phase 4 clinical trial, which cryo-EM have been applied, is increasing, and also we see a number of approved drugs in which cryo-EM has played a significant role, like the COVID vaccine, like the RSV vaccine, for example. That is a little bit about the introduction that I would like to give. Of course, a lot of instruments play an important role in getting all these data. And one of the instruments that played a significant role already since the early days in the development of single particle analysis and tomography is the Krios microscope. The first version was launched in 2008. Some of you might still remember or have that instrument. In 2015, the second generation was emerging to the market, followed by the third generation in 2017. In 2019, we introduced a system which was smaller from appearance, especially from the height, smaller footprint, also a different look and feel. That was the Krios G4. And now 2025, we release the next generation Krios instrument, which is called Krios 5. Now, what is actually the Krios 5 about? And not only the Krios 5, but also its previous generation. It's all actually about improving productivity and resolution. And as you can see in this particular slide, the Krios has really played a very important role, thanks to also the development of more intelligent software, of faster camera, direct electron detectors in particular, but also energy filters and cold-FEGs. Together, it has actually led to a significant improvement in throughput, number of images per hour, and these are of course single particle data. And simultaneously with that, also the resolution has been significantly improved. As you all know, we touched the atomic resolution barrier, or crossed the atomic resolution barrier in 2020 with a groundbreaking paper, and with let's say a significant amount of structure, we're really touching more and more routinely towards the 1.5 and 1.2 angstrom resolution in a very short time. If you talk about acquisition times, basically you can say in 2015 we spent 50 hours on acquisition time to get to 3.6 angstrom resolution, and now we can only need 6 hours in order to get to 1.2 angstrom resolution. So with Krios 5, we'd like to extend that trend. We'd like to continue the improvement with respect to productivity, resolution, and of course also ease of use. In the next couple of slides, I will talk further about the specific new benefits that Krios 5 has for single particle analysis and for cryotomography. First couple of slides I will spend on single particle analysis. There are a number of benefits for that technology that are unique for Krios 5. First of all, we have new AI-assisted smart EPU plugins. I will talk a little bit about that in the next couple of slides. We have extended aberration-free image shifting, pushing the resolution further, and we have improved the basic integrity, if you like, and accuracy of the instrument by improving the magnification calibration accuracy within 1% range. But first of all, As a kind of benchmark example, we show you this lye protein, which is a protein that plays a role in the development of sugar substitutes, as well as the synthesis of antiviral drugs and other potential therapeutics. And we managed to get this image reconstructed at a 1.52 angstrom resolution in a 25% faster time than the G4 as a benchmark. So that already shows that same quality can be obtained, but then significantly faster. And I will tell you why that happened. It has to do with all these new benefits that I summarized on this slide. The first one being, as I mentioned, the smart EPU plugins. Well, EPU has been largely automated the last couple of years, from the recording of a grid atlas all the way to the acquisition of images. And of course, there are a number of crucial in-between steps that are important for successful performance of a single particle run, like the selection of the best grid squares, like the identification of foil holes, and like the selection of the best possible ice quality areas. Well, thanks to AI-driven pre-trained neural networks, we have been able to, let's say, automate and make the central part of the screening process more intelligent. So the selection of the grid squares, identification of the foil holes, and also the identification of the best ice quality areas has been automated. Microscope gives you an advice. You don't have to, of course, comply with that advice, but the microscope gives a pre-selection of those best areas based on looking to contamination, brokenness of grid squares, for example, aggregation artifacts, stitching or detection issues. That's all automatically, let's say, taken into account to this neural network training. So this entire smart screening improvement increases obviously the efficiency because less time is needed to exclude bad foil holes, for example, but it also enhances the adoption because you don't need the expertise to do everything manual. You still can, of course, but you don't need to do that anymore. And of course, in the end, it contributes to higher data quality because you should only select the best possible areas. Then another benefit that we added for Krios 5 is the aberration-free image shifting, and then the extended version of this one. I would like to go quickly back in time to the time that we talked about the first 3 Krios generations, as I showed in the previous slide. In those days, when you wanted to move from one holder to another mole, you had to use apply mechanical stage moves. And when every time a mechanical stage move is made, there is a relaxation time involved, obviously, because the stage is not directly, let's say, standing still the way you would like to do it. So you lose some time as part of the settling time process. Now, in 2019, with the introduction of Krios G4, we introduced aberration-free image shifting, which basically allows us to do image beam shift movements up to 12 microns. And when doing that, there is less or no relaxation time. And that means that you can speed up your acquisition process. We managed to do this while correcting the coma, which happens when you go to 12 micron tilt. And that was one of the main drivers for the enhanced productivity that I showed you in my introduction slide. What we're doing now with Krios 5, we're introducing extended APHIS. So we go one step further than what we did with APHIS. We can now go to 20 microns image beam shift without introducing aberrations. We learned that when you go to 20 microns image beam shift, you introduce a second order aberration correction on top of the first order aberration correction, which is typically linear, but we have modeled this and we can fully correct for this. And in addition, we have improved the targeting accuracy, which is getting increasingly important when you want to move to these very extreme wide ranges with the image beam shifts. Last but not least, we also know that there is no, let's say, sacrifice on the image quality. So we enhance throughput, the quality remains the same. And that is exactly what I showed you in the introduction slide of the single particle part, where we see that we have 25% higher throughput. In this case, we can go to 930 images per hour compared to the 740 images per hour. That is the benchmark study on Krios G4. But that's a very important driver for enhanced productivity. And of course, it depends a little bit on the use case. Typically between 15 and 25% throughput can be gained with this extended aberration-free image shift option. The last new feature that we introduced for single particle analysis is actually an improvement of the magnification calibration accuracy. And this, of course, is quite important because for predictable and reliable prediction of a 3D model, it's important that the magnification calibration has been done as accurately as possible. Now, in this case, we went to use a different ground truth sample. We use now the silicon 110 sample to measure the lattice spacing more accurately based on FFTs. That's a surface action, but it allows us to determine the correct pixel size within 1% accuracy. And with that, you basically create a much better starting point for more efficient processing. And as you can see in this slide, with the CTF behavior is much more aligned when you go to 1% magnification calibration accuracy compared to 5% magnification calibration accuracy. It can still be improved further, of course, below 1%, but as I said, the starting point is much better at this particular specification. Then the second set of slides, I will talk about the improvements for cryo-electron tomography, which is, as I showed you, becoming a really technology that is on the rise. We have three new aspects or benefits that we would like to introduce. The first one is on-the-fly motion correction, but now in combination with 8K decoding in ER mode. We have improved the possibility to enhance the quality of the lamella that is created in the Arctic by using a vacuum capsule that is used for transfer between Arctic and Krios. And we have made some significant advances in the tomography software, both on the setup part as well as on the acquisition part / throughput enhancement part. I'll first talk about the first benefit, which is motion correction in combination with 8K ER data. Motion correction obviously is very important to be corrected for, especially for tomography, where we take movies at very stilt angles, and of course the frames need to be aligned in order to get to the best possible resolution. So for subtomal averaging, this of course is also very important, and that is exactly one of the applications where this 8K motion correction option can play an important role. As you can see in the example on this slide, you can be limited by physical Nyquist, that you cannot go beyond, let's say, the Nyquist frequency when you use the 4K mode, whereas in 8K you can benefit from the subpixel accuracy localization beyond Nyquist, and that pushes the resolution further in the subtomogram averaging regime. On average, the resolution is 3 angstroms in this particular example versus 3.9 angstroms for the 4K example. You can choose in the tomography software whether to use 4K or 8K. Depends, of course, a little bit on your use case. If you go to lower magnifications for tomography, for example, larger fields of view, 8K may be beneficial. And if you use subtomogram averaging, like I said here. For somewhat higher magnification applications where you would like, for example, to look to membrane parts or details of cell, 4K is good enough, and then 4 can be selected. Important maybe to mention that although the file size of the 8K sample is 4 times larger than the 4K sample, there are no time penalties when you use 8K versus 4K. They're equally fast in both cases. Another very important improvement is related to the maturity of the tomography workflow. In the current situation, typically, and this is one example, of course, you grow cells on an autogrid. On a grid, you vitrify the grid, put them into the autogrid, put them into the cassette of the Krios, into the capsule, and then you load it to the Arctic, in this particular case, to prepare lamella. And once the lamella have been created, the samples are transferred in the standard capsule to the Krios, and then the tomography experiment is started. That's the typical workflow that you currently have. However, we've noticed, and I will show you that on the next slide, that there is quite some contamination risk on the lamella if you have the sample inside liquid nitrogen. And we would like to avoid that. And for that reason, we've now come up with a new piece of hardware, a new capsule, a vacuum capsule, which allows for the transfer of samples under vacuum conditions from the Arctic to the Krios. And by doing so, there is less contamination, as I will show you. How does that work? And this is just a very short movie summary of what will happen. This is the vacuum capsule. The capsule is pumped vacuum, by the way, through the autoloader in the Arctic or in the Krios. In this case, pretend that samples have been vitrified with, for example, with the Vitrobot, put into the cassette, transferred to the Arctic, lamella are prepared, and once that happens, and has happened, and that's what you see in this video, you go to the Krios microscope, and under vacuum conditions, obviously, like I said, the vacuum is pumped through the autoloader, and then the vitrified samples and prepared lamella are loaded into the Krios. The cassette is uptaken with the vertical axis into the autoloader, and the grids are selected for setting up the tomography experiment. Basically, it's the same way of working as with the standard capsule, the non-vacuum capsule, but the results are significantly better. As you can see on this slide, on the right side, you see something that could happen if you use the standard capsule. There's quite often contamination inside the liquid nitrogen itself, but there can also be contamination, of course, from water in the air, and that then gets condensed onto the liquid nitrogen and to the lamella. So small crystals, big crystals, ice network type of structures, all things that you don't want to have in your precious lamella, obviously. And if you look to the vacuum transfer result or the vacuum capsule result, it looks much more clean. And so if you would like to select specific areas from the lamella for your tomography acquisition, then of course you benefit much more from a clean sample. And that is what you can see. And this is something we see, let's say, repeatedly. Of course, this is statistically validated. And we talk about the tomography workflow. It's not only about the hardware and connecting hardware in the most optimal way, but there's also software involved, and software gets increasingly important, obviously, as I already showed in the single particle part of my presentation. Also for tomography, we've made significant steps to enhance the setup of a tomography experiment and the acquisition of experiment, and that is what I would like to talk about in the next part of my presentation. So with respect to the tomography software, we are introducing what we call the Cryo-tomo suite. And the Cryo-tomo suite consists of some new acquisition routines, you can say, features, functions, like the task parallelization. On my next slides, I will talk a little bit more about that. The Tomo multigrid, equivalent of EPU multigrid, is what we're introducing here. And there is a nice possibility to analyze all the results that you have been made and look in one snapshot overview what you have been doing in a discovery viewer. In addition to that, we also have this 8K motion correction option, which I already talked about. It's a button in the Tomo software that you can select. And we have also automation of specific routines, actually, that you would like to use. For example, fine beam, auto-stigmate, auto-coma. The routine alignments have been automated, and they can also be found in the application software for tomography and for SPA. But first, about some of the new highlights: task parallelization, Tomo multigrid, and discovery viewer in the next couple of slides. Task parallelization is all about enhancing the efficiency of your tomography experiment while you set it up. So while acquiring a grid atlas, and that is what you see happening on the movie on the lower side, one can already start thinking about defining the search maps at a higher magnification. Basically, looking to the best possible lamella that you have created in the Arctic. That's one synchronization step. The other synchronization step is at higher magnification. When you want to zoom into the acquisition of a search map, you can already start defining the batch positions, or the best areas where you like to take, for example, multi-shot tomography of. Or you can also continue defining more search maps. You have the option, of course, what you would like to do, but also here an important parallelization step can be done. And even while you acquire tail series, you can continue defining batch positions. So this is actually very, let's say, convenient. Of course, it's still a manual process. It's not that like for smart EPU, we have smart Tomo and the system automatically tells you these are the best lamella, and this is the place where you can, where you should do the batch Tomo series of. So this is still manual, but it's very efficient in terms of the way it is set up, and it all fits in the enhanced throughput of the tomography workflow. In that story, there's also a Tomo multigrid that plays a very important role. So the process that I just showed you on the previous slide, where you go to a grid atlas, you zoom in, select the best possible lamella, again go to high magnification and take basically determine your acquisition scheme. So we call it multi-grid, multi-site, multi-shot. We can now do for multiple grids, basically for an entire cassette that is loaded with auto grids. And of course, that enhances the efficiency again significantly. What is also very important, and that is also why we've implemented this, is that it increases the throughput, because there is no manual interaction needed when switching grids. You can just run it for days in principle, depending on how you've set up your experiment. And this slide basically shows an example of how that would look like in reality. So there are some parts that the microscope, of course, is taken care of in an automated fashion. That's the red part, the run part, and the user interaction is mainly limited to the first part of the tomography experiment, where you do the setup phase. And that goes hand in hand, as you can see. That's the parallelization I was referring to. So especially when you use Tomo multigrid, then you can collect multiple atlases of all the grids in a cassette. You can in parallel already define search maps, as I mentioned in my previous slide. And while acquiring search maps, you can define already the batch positions. And that is, again, a very efficient thing to do. I have to say, of course, it can be quite tedious to set up an experiment, because it can be, you can basically, especially when you do Tomo multigrid, define a lot of experimental areas. So we have the possibility to pause and resume the setting up phase here. So flexible management of microscope time in that respect. But once everything has been set, you can click on the batch acquisition button, and there's an unattended high throughput acquisition of the tail series that you defined, and also the exchange of the grid, as I mentioned, can happen automatically, and the relocation to the specific targets. And that can happen for up to a week. So in this particular example, it's a recording of over 1,000 tomograms, but of course it can also be less or more, depending on how you define it. Now, the last new thing that I would like to mention here in the tomography software is the discovery viewer that we're implementing, which basically brings all the words together. It runs in CryoFlow, and it allows you to look at all the atlases that you've taken, to all the specific search map that you've acquired, to zoom in to the specific batch position, the multi-shot position, and then also show the tomogram. So for a selection of quality of the best tomogram, for example, you can go through this discovery viewer and interactively select and play with all the experiments that you have done, including having insights in also the quality metrics data, like astigmatism, defocus values, etc., etc. So that is with respect to tomography. The last part of my presentation, I would like to talk a little bit about a complete new aspect, but a very important one, and that is sustainability. And we all know that electron microscopes are very energy consuming. As a rough rule of thumb, the average energy consumption of an electron microscope in the Krios is around 35 households if they consume 2,500 kilowatts per year. So it's important, we thought, to cut down the energy costs. And that is what we have done by introducing energy saving eco modes. On the next slide, I will talk a little bit more about it. In addition, we are also having an environmental impact label, an ACT label, which was given to us by an accredited independent third-party company, My Green Lab, who investigated the instrument's footprint, the manufacturing process, the materials that were used, the recyclability of the materials, and based on all these numbers and scores, we got the accreditation for the ACT label. But what saves the energy? That's actually, I think, the most interesting part here. Those are the following two options. So what we can do, we can go put the microscope into a sleep mode. That's a very deep sleep mode. And we have a second option, which is somewhat lighter mode. We call the standby mode. When you go into sleep mode, you switch off significant components of the microscope that consume most energy, like the lenses, the deflectors, the electron optics, the correctors, sigmators, and the chiller. And by doing so, you save up to 84% of energy compared to being in normal standby mode, which is what you would do if you would now have, for example, a weekend break or a holiday break. Of course, an important implication here is that you would go for a pause in your microscope acquisition mode. So suppose you go for a Christmas break or long holiday break or weekend break, that is the moment when you would like to activate the sleep mode, for example. This activation happens in a specific software piece that we've designed that also contains some of the automated alignments, the microscope companion mode. And there you can determine when you would like to put the microscope into sleep mode, but also when the system needs to slowly wake up. Because typically the recovery targets, the recovery time is around 12 modes for the deep sleep mode. So suppose you would like to start working on Monday morning again, 9 o'clock, you tell the microscope, Sunday evening at 9 o'clock, please slowly wake up. Important side comment is that the alignments remain stable. They don't need to be redone when going into those sleep modes. Alternative mode is the somewhat lighter sleep mode, which is called standby mode. And that is something you would typically consider to use if you, for example, have an overnight break somewhere and you only switch off some of the detector modes and the TMPs potentially. That saves you less energy, 5 to 10%, but on the other hand, you're within an hour back to standby mode. So those two options actually exist, are new. And when you go into sleep mode, the microscope also gives a specific indication. We turn the LED lights into green, as you saw on the opening slide of this presentation, indicating that you're saving energy. Alternative LED coloring is still available. The cold mode is blue LED light, and the warm mode or cryocycle mode gives you the white LED lighting. So that summarizes the Krios as I've presented it to you. This slide shows roughly in a summary snapshot mode the main highlights. So we have improved and contribute to high data fidelity with improved magnification accuracy within 1%, improving not only data fidelity but also the throughput by having extended AFIS as a feature, and we have implemented routine alignments to make things easier in the setup phase. We have enhanced the efficiency of the workflows, both single particle and tomography. And when we talk about tomography, as I mentioned, we have the introduction of the parallelization routines, the Tomo multigrid, and the discovery viewer that plays an important role. We are leveraging more the potential of the Falcon for eye camera by introducing 8K on-the-fly decoding for larger field of view and subtopical averaging experiments. We are introducing a connection, optimal hardware connection between the Arctis and the Krios for robust and contamination-free transfer of lamella by introducing the vacuum capsule. And last but not least, we are taking the first steps on providing a sustainable solution for the scientific field by providing energy-saving eco-modes and by having this My Green Lab Act Eco label. Thank you very much for your attention. And before we take your questions, we would like to show you a video which we created that shows in more details the tomography workflow that I explained already in my presentation, and all the essential steps that you need to take from a scientific point of view. So enjoy the movie, and after the movie, we'll happy to answer your questions.
Thank you, Mark, for this great presentation. It's exciting developments for both single particle analysis and for cryotomography. We're now going to have a deeper view of the workflow to lamella generation from the Arctis plasma fib to the Krios 5. Alex Riggett works as a product marketing manager for Thermo Fisher Scientific. He developed and used cryo-focused ion beam instrumentation for applications in electron tomography at the Max Planck Institute of Biochemistry in Munich and has more than 15 years experience in cryo-electron microscopy. Alex is going to take us through this workflow, and we're very much looking forward to seeing the details of how it works. Alex, over to you.
Yeah, thanks very much for the nice introduction into the new Krios 5. My name is Alex, and I would like to show you how the Arctis cryoplasma fib system can be utilized to produce samples for the new Krios 5, and how the combination of both instruments can streamline your tomography workflow. A very critical step in the cryotomography workflow is the production of thin cellular samples for the cryotransmission electron microscope. And the microscope that we utilize to produce such samples is a focused ion beam system, or in our case here, the Arctis cryoplasma focused ion beam system. Marc already showed you in his previous talk how the vacuum capsule enables us to do clean transfers of this very delicate cryolamellae into the cryos. So that's an enormous advantage because we can avoid all transfer contamination that can be picked up by the samples. Other developments which are very crucial are listed on this slide, and this is on the Krios 5 side, the tomography software improvements that Marc already mentioned in the previous talk. But also on the focused ion beam side, there's on the Arctis, there's essential software which allows us to automate the lamella production process. And we can do that not for one crit, but for up to 12 crits with the robotic loading system that both of these systems feature. Yeah, this slide shows very nicely the power of automation software for the production of cryolamellae. Here we used the new automation software called WebUI on the Arctis cryo-PFIB system to produce multiple lamellae on a crit. In this example here, over 40 lamellae that have been produced with the automation software and which can then be transferred over to the cryos. I will go into a little bit more details on how this new automation software is utilized on the Arctis in the upcoming slides. Before I come to the WebUI software, I would like to make you aware of another important development that is enabling multi-crit automation workflows, and which is in turn very important for cryotomography, and that's the TomoCrit carrier. So whenever you want to do workflow automation, whether it's on the CryoFIB instrument or on the Krios instrument with Tomography 5 software, as you heard that in Marc's talk, it's very important that you maintain the orientation between the tilt axis in the TEM and the milling axis in the FIB instrument fixed to each other. And the TomoCrit carrier is a carrier which can do this. So because of the flat sides, it's always sitting in a fixed orientation in the Arctis cryo stage, as you can see here on the example, as well as in the Krios Compute stage. So that enables us to load and unload crits, as you can see in the lower panel. So here we have loaded and unloaded crits, the same crit, multiple times with the robotic loading system of the autoloader. And in each case, the tilt or the milling axis here remains constant. So this is truly enabling multi-crit workflows and a very important development for cryotomography. You heard already about the new vacuum capsule, which provides clean cryolamellae transfers into the Krios 5. I would like to show you now the WebUI automation software on the Arctis system, which is another very important development for the streamlining and automation of the cryolamella production workflow. The basic idea of the WebUI software is to provide a single software for providing the entire automation workflow for cryolamella production. In that sense, WebUI is unifying tasks from 4 different existing software pieces. You can see that here. So this is IFLM software, MAP software, AutoTemp Cryo software, and the system control software XT. So all functions from these 4 software pieces are unified inside WebUI. So that means from a user perspective, you only have to use WebUI as the central software for the automation of cryolamella production. The unified single software concept brings certain advantages. In this case, we don't have users sitting next to the microscope to work on their samples. The samples can be loaded via the autoloader into the microscope. The microscope can carry up to 12 samples. And it's possible to access the WebUI software remotely. That means from any web browser within the local network or via VPN, you can always access the WebUI software to set up automation runs. And since you can load multiple samples, also interesting for multiple users who may share samples on one of these autoloader cassettes. So it makes it in a way a perfect system for facilities because of these multi-crit capabilities, and because of the user-friendly web UI system that allows multiple users to interact with the system. So when you open web UI in the web browser, you enter an IP address, and then you get the login screen, which looks like this. You log in into the system, and that brings you to the main user interface where you can set up the automation runs. The user interface provides all different types of information. One is, of course, also a status page where you can always get information about the system, the disk space available, the status of the autoloader and the chamber. Particularly important here to see the filling levels of the liquid nitrogen, how long it takes, for instance, to the next refill cycle, and what are the temperatures on the essential parts of the system. So that's kind of information which is provided in the status page on the user interface. Web UI is using a grid-centric approach for setting up automation runs. That means you can load up to 12 samples into the system within the autoloader cassette. And on the grids tab that you can see here, these loaded grids will be displayed. So you can see number 1 until number 8 on this screen. For each grid that is loaded, you typically would start to acquire first an overview image. You can see these overview images displayed here. These images allow you to quickly judge the quality of the grid. It's of course very important that for tomography experiments you have an optimal prepared grid for the lamella production. So this gives you a quick assessment on the quality of the grids. Then for each grid, you can define a certain actions in this grid-centric approach. You can set up tile sets to define the actual lamella sites so that are higher resolution images of the grid, where you can select cells from which you want to prepare lamella sites. And of course, you can define the lamella production parameters starting from the protective conductive coding steps with the GIS system and the platinum sputter target down to the ways how you want to produce lamella. So all this you can define in these steps here for each of the different grids loaded. With the tileset option, you can obtain different images at a higher magnification of the grid central area, so that allow you to select regions for lamella sites, which are marked here with the crosshairs. And there are different ways to obtain such tile sets. So in this case, you can see this is an electron tile set taken with the electron beam, but we can also take different tile sets, for instance, with the ion beam. The ion beam also produces a secondary electron image, but it provides more topographical information about the grid. So you can now see a little bit better how the cells are distributed, because you look from an angle onto the grid. You can nicely see how the cells are distributed unevenly across the grid. And in case you couldn't identify cells for lamella milling properly on the electron tile set, the ion tile set provides a different imaging modality to select lamella sites for automation. So you can see the sites which have been already marked here. They contain numbers, and these are the small rectangles basically define the sites where the automation software will produce lamellas. Yeah, a third option for this grid overviews is to use fluorescent light microscopy information about the cells. And this is done by recording a so-called optical tile set with the integrated light microscope of the ARCTIS system. You can see how such a fluorescent map of the central area of the grid looks like. Here we have two channels selected, green and blue. Blue corresponds to the nuclei, which are stained here, so you can see the distribution of the cells across the set across the grid in fluorescence. Of course, you have also the capability to use reflection images or also other channels to complement information. This is very important if you look for a sparse label that you want to identify, or a certain phenotype which is fluorescently tagged. So then this type of optical tile set gives you a good way to spot for possible lamellar positions. The lamellar process is automated, of course, and you can see here on this image a final outcome of such an automation run. So this is the final lamellar produced with the automation software. You can also see some of the features that were defined in the lamellar milling template, such as the production of the stress relief cuts to take out tension in the vitreous sample here, as well as the milling of cross fiducials, which can help in cross correlation processes that you need to set up the automation, and as well as for the light microscopy targeting. For each lamella produced, there's a way to rate the lamellas so that you can also kind of highlight which is a very good lamella or immediately and retrieve it later in the TEM. All the individual steps of the lamellar production process are stored by the system, so you will have also access to these images once the automation run is completed. A nice feature in the ARCTIS Web UI software is the targeting function, where you can use light microscopy information to guide the lamellar milling process. It is shown here. This is showing you on the right-hand side here a reflection image taken with the integrated fluorescent light microscope, overlaid with two fluorescent channels, so the green and blue channel. In blue you can see here nicely the nuclei, in green some mitochondria which are labeled here. And we have implemented the targeting workflow where you can go through Z stacks of the sample taken with the light microscope, define the feature of interest within this 3D Z stack information, and then use that information for targeting the lamella in the automation software. And the final outcome should look like this. So it's an image before the lamella has been produced. And we can see here the features that have been targeted are still present in the final lamella. So here's an example how the fluorescence-based targeting for lamellar production works. Here is a light microscopic data set. You can see the different fluorescent channels here. We can adjust the histograms. And more important, we can go here through the Z stack with that slider, so that brings the cell into focus. And for the targeting, very essential to place a fiducial marker here, and then relative to this fiducial marker, we look for the sharpest focus plane in the fluorescent data set, which defines the position of the feature that we want to target. And now we can move over to the SEM, or to the ion image in this case. And we simply utilize or retrieve the fiducial marker again. So this image on the left is the integrated light microscope, the reflection image, which of the fiducial marker. And by selecting a corner of the fiducial marker, this is also retrievable here in the ion image. And by moving this yellow crosshair to this marker, also the orange region of interest marker will move, and that indicates then the region of interest for the lamellar targeting. Yeah, here's again an overview image of the grid, and this shows you how you can set up multiple sites, multiple lamellar sites on such a grid. We can browse the grid, look for cells which are in a favorable position for lamellar milling, for instance here in the center of a mesh. It's also possible to obtain sparse tile sets with the light microscope, so you don't need to do the full tile set, but you can also select certain areas where you want to take fluorescence information. Here are 2 areas marked. And beside this, you define the lamellar sites fairly easy by clicking on the cells that you want to prepare. You have a way to select different milling templates when you make these selections. All these sites are queued in the system, and then the automation software is processing the sites and producing the lamellas in an automated fashion. So this is a relatively straightforward way to set up 40 lamellar sites here. All of the sites that you have defined, they will be scheduled into a queue. And you can see also the parameters that you have set for each of the lamellas. There are different templates that you can choose and tune and select. Depending on the template, the steps that are done are kind of defined in the template. So for instance, some of the lamellas will contain a 3D targeting step, others maybe not. So there's freedom to design for each of the lamellas different templates. Again, once you've set up the lamella site list, it will be queued, and for all of the sites that you defined, there is this list made. The system also will tell you once the preparation steps are finished, once the coarse milling steps are done, and once the final polishing steps are done. And in case of success, the system shows it as a finished green lamella box here. Yeah, I showed you before how you set up individual sites for lamella milling. Once these sites are ready for the milling process, the system will ask you to define the milling boxes, and that's what you can see in the video here. So here we have a couple of lamella sites that were previously selected, and as a user you now select where you want to exactly place the boxes across these cells. Once you're done with that, the automation software starts. So this is a look at the underlying system control software, XT, which is not displayed in the Web UI software. Here, that's what's basically happening in the background. The system is doing a multi-step approach to produce the lamellas, starting with a high milling currents in the first milling steps, and then going from the coarse lamella production in steps down to final polishing of the lamella. You can see here cleaning cross sections which are applied at lower milling currents until we have produced the final lamella from the sample. Yeah, once the lamella run is completed, the system, the Web UI software will display you a gallery where you can inspect the lamellas produced. There will be all the parameters stored for each lamella. You can rate the lamellas. Some lamellas will be good, others may have some damage on it. So you have a quick way to judge which lamella is well suited. And all this information can be also retained then when you go into the TEM. So when you import data from ArcGIS Web UI into the Krios 5, you can get already the information where are the good lamellas from that grid that has been produced, and which are the lamellas which you probably want to omit in the TEM tomography. With this, we are back in the tomography workflow that you can see on this slide. So just remember, we produced cellular samples. We had cells that were grown on EM grids, that were frozen in the Vitrobot device, then loaded via the autoloader system into the ArcGIS plasmafib system. And in the plasmafib system, we have produced now a number of lamellas. That means we are now ready to transfer the samples into the new Krios 5, where we obtain the tomographic data information. Yeah, here's an example how cryo-lamellae that have been prepared with the ArcGIS system look in the transmission electron microscope. These were rather thick chlamydomonas cells which have been thinned with the focused ion beam system down to less than 200 nanometer thickness, and which are now ready for tomography data acquisition. Yeah, the new Krios 5 also features, of course, the latest version of our tomography software, Tomography 5, and this allows you to obtain multiple tomograms per lamella. Here's an example where you can see 13 different sites that have been set up with Tomography 5. The 4 sites which are highlighted here, the tomograms are shown on the right-hand side. In addition to this multi-area acquisition, there's also Tomo Multigrid, which allows to image multiple grids. This is, of course, a feature which benefits a lot from the Tomo Grid carrier and then really enables multi-grid workflows. And last but not least, there's Tomo Live that has been mentioned before also, which provides a live reconstruction of the data as it is recorded. With this, I'm coming to the end. I would like to thank you for your attention. And I hope that I was able to show you how the ARCTIS cryo-PFIB system, together with the new Krios 5, can streamline the cryotomography workflow. Thank you.
Fantastic. Thanks both to Alex and Mark for these great presentations. And we're now going to start with the Q&A section of the webinar. We've actually had loads of questions that have come in, but if you think of anything else during the course of the Q&A, please continue to type the questions and we'll address as many as we can. Any that we don't get to, of course, we can reach out to you by email after the event. So please continue to type your questions. I'm pleased to be joined for this part of the webinar by two additional members of the Thermo Fisher team. John is a Senior Product Manager for CryoFib and SEM, and Wright is a Software Product Manager for cryotomography. So we have a great panel to be able to address your questions. Maybe if I start on the Krios side of things, Mark, will it be possible to modify a Krios G4 autoloader to work with the vacuum capsule? Question mark.
Yeah, that's a good question. And the answer, yes, that is possible. Although the autoloader requires some modifications. So especially because currently it doesn't allow to pump the vacuum capsule because we have a normal capsule. We need to make some adjustments, and that will, yeah, all be part of a potential functional upgrade package of the autoloader and of the Krios G4 microscopes in general, which we'll make available in due time. But yes, it is possible technically, and we will make it work.
Fantastic. Okay, thank you. Another question on the vacuum capsule, so I'll stick with you. Can the vacuum capsule also be used to store samples for longer periods of time?
Yeah, that's a very good question. But the vacuum capsule has been designed as a real transfer module. So, like Alex nicely explained, going from the Arctis to the Krios, there is a standard hold time of roughly 20 minutes. So you can take it easy. If your microscopes are not in the same building, you can nicely walk from one building to another without the risk of having additional contamination. But it's not intended as a storage medium to keep the samples in for a week or so. That was never the intention.
Okay, great. Thank you. That's clear. John, maybe I could come to you for one. How was the test of cryolamellar contamination done? Same lamella with and without contamination. And do you have a contamination test protocol that you could share?
That's a very good question, and of course, yes, it is difficult to measure when you go from the Arctis to the Krios in that order. In this case, to illustrate the effects of liquid nitrogen, we did the forward transfer where we had the clean lamellas first, and then we unloaded it into liquid nitrogen and then put it back to show what the transfer into a plan 3 or the regular capsule contributes. And so that way you can see the before and after, if you like. So we were simulating it in reverse in this case.
Fantastic. Thank you. Okay, right, let's come to you. There's a couple of questions on the acquisition speed. How come acquisition speed is the same for 4K and 8K? And also related to the same kind of topic, can you do 8K in SPA?
Yeah, two very good questions. Thanks for that. So basically, our EER format that we're using on the Selectris Falcon 4 combination is acquiring data at higher pixel sizes, and we basically use now all the available data in a way that we didn't before. So yeah, basically it's more of a decoding step that we now make more easy for users to leverage. And this gains some benefits, but for SPA, basically you don't really see that much benefits for using 8K, because the benefits for using 8K are more on the TOMO side of things. So if you're prioritizing a larger field of view, say go for a 2 angstrom pixel size or above, and looking at macromolecular complexes and do subtomo averaging. So even if you're doing tomography with a 1 angstrom pixel size, there is not really any benefit to go to 8K. You can just use 4K and get the same good results. So therefore, to make it easier, we said, okay, we just keep SPA at 4K and only introduce 8K for those use cases where it actually makes sense. And that's the same thing if you don't do any subtomal averaging, then also 8K doesn't really add any value.
Okay, fantastic, great. Mark, there's a few questions on the green aspect. So after sleep mode, is the calibration preserved? And also, is it possible for regular users to wake up a sleeping system remotely? So 2 questions on the sleep mode.
Yeah, very good questions actually. And I assume that when you say calibration, you mean alignments. Are the alignments preserved when you go from sleep mode and get back into the activation mode? And the answer is yes. We thoroughly tested it, and there is no need to redo alignments after you wake up. And so that period is, as I mentioned in the presentation, 12 up. If you go from deep sleep to activation mode, that the alignments are as good as they were before you enter that sleep mode. And talking about controlling sleep modes, we deliberately made the sleep mode active actually or accessible for supervisor users, not for regular users, because it is quite a significant step that you activate in the software because you switch off the electronics, the chillers. So we don't want the regular users to mess around with that accidentally. So the supervisor can only activate the sleeping mode. We have a standby mode as a second option that can be controlled by normal users.
Fantastic, thanks, Mark. And then there's one more on the energy saving. So will the Krios 5 also save energy when in operation mode?
Not yet, but it's an excellent question because after all, you buy a microscope to use the system. So it would even be more efficient if you can save energy while in use. It's something we are looking into. It would require probably new electronics because the new electronics are more energy savvy in general. So it would require a significant redesign of the system. But it's definitely a very important thing that we are looking into as a phase 2 in our sustainability efforts.
Fantastic, thanks, Mark. Okay, something on the tomogrids. Can you briefly explain what adjustment of a Krios autoloader is needed to be able to handle the tomogrids, i.e. can this be done for older Krios? John, I'll come to you with that one.
So yes, it can be done for certain generations of autoloader. I will say that's for a G3 and a G4 is certainly possible. What we're doing there is we're aligning the handover position from the cassette to the first step in the final alignment procedure, and that requires some special tooling for the service engineers to adjust the position so it's really precisely centered, and that gives us then the ability to load it with the reproducible positioning that's needed for the tomogrid alignment. That's also one of the reasons I think, just to tackle the timing, I see the timing question comes up quite a bit. It will be on Krios 5 and Arctis systems later on in the year, coming out of the factory. And of course, we'll have some rollout to the field depending on the availability of the service kit, because it needs a physical suitcase type alignment device that we need to get into the field and then roll out the alignments there. So that will come out during the later half of this year and early next year. Systems.
Brilliant, thanks, John. And then on an automation question, one for you, I think, right? Regarding Tomo automation, are there any plans towards smart, as in AI-assisted, target selection in Lamelli?
Oh, that's a wonderful question. This is something that we have on our roadmap, and that we get requested quite a number of times. It depends a bit what direction to go into, because there is a lot of attention right now in making it smart and AI enabled. But leveraging, for instance, already the data that comes from Arctis, the fluorescence data on a search map in Tomo, also goes a long way in helping people. So it might be smart as in AI enabled, it might be in a different way, might be combined. We definitely have a lot of plans in that direction.
Fantastic, thank you. Mark, back to you for the green topic. So talking about energy and sustainability, how can we plan for recycling after service instrument part renewal, for example, to recycle parts of the vacuum pumps, for example?
Yeah, that's an excellent question, and that is also related to the ACT label that I also mentioned. As you have heard, we have this Act Green label for the Krios microscope, and that not only entails the energy saving mode, but more importantly, how do we as a company handle recycling of components, but also packaging of materials. All these things actually are taking into account into the equation. So with that, if you look to the Act label in detail, you see that some things are green, some things are orange, some things are red. The red things are improvement points for us. And with respect to recycling of parts, for example, that's also one of the things, I think we score yellow there, that we need to take more serious. So taking components back, taking packaging material back, recycling things, that's definitely one of the things that we want to boost more as part of, let's call it sustainability 2.0. But these are the first attempts that we now put into the market. But there will be more to follow, including recycling.
Great, thanks, Mark. Good to know. We have a question on fluorescence targeting. John, for you, can you explain how the fluorescence targeting works inside Web UI?
Sure. Actually, the fluorescence targeting is a multi-stage process in ArcTIS, and you have essentially 4 different imaging steps as you go through a workflow, which you can optionally turn on or off. So in the first case, you would have a tile set overview to help you identify the right cells, the right locations. And that can have different settings, say, from the next step, which is then once you've chosen those cells to do maybe a high resolution stack with on a particular cell to help you with the 3D targeting. Of course, obviously, when you're milling the cells, it can move. So then you need to maybe do another targeting step to refine the coordinates. There's an optional one during the chunk milling stage. And then, of course, at the end, there's another step where you can take a final fluorescence image as part of the flow to confirm the fluorescence that's still in the lamella. There was a question on that. And, of course, all of these are independently adjustable for power and things depending on what you would like to do. So that's all part of the workflow.
Thank you, John. Okay, so software developments now, right? One for you. Will the software developments, like the mentioned Smart EPU or Tomo Multigrid, be available for the Krios 4 without necessitating the upgrade package?
In principle, yes. So Smart EPU and Tomo Multigrid will be available. They are actually already available. And especially the Smart EPU improvements, if you have a current service contract, will be included. Multigrid in Tomo is an additional license, so you need to get in touch with your account manager to arrange how to get a license for that. But in principle, any system that supports Tomo 5 will also support Tomo Multigrid. And I wanted to add something related actually to the Tomo grid questions that were asked before. What is needed in terms of alignment? One thing that I wanted to stress is that while on ArcTIS you actually do need to have a Tomo grid to enable multigrid and use it to mill in the proper way. In Tomo Multigrid, we actually do allow users to use autogrids for acquisition. We do realignment when changing grids, and we get really good results. Of course, if there is some rotation, that can cause problems with your missing wedge, but in general, it seems that the results also with autogrids actually are pretty good.
Great, thank you very much. We have a few questions on kind of room requirements and dimensions. So Mark, maybe if you could take us through what are the new dimensions of the new microscope and the room requirements?
Yeah, they're actually exactly the same as the previous microscope generation. So there is no, we reduced the height, as you know, for when going from Krios G3 out to G4, 2.97 meters, room requirement 306. That's the same for Krios 5. You don't need to do additional room renovations to get to host the Krios 5.
Okay, fantastic. And back to Lamelli. John, is the transfer of Lamelli to grids automated? And also a question on Web UI, does it recommend which areas are best to mill?
So we're not transferring the lamella in Arctis to the grids because we're milling the lamellas in situ. So in this case, whether you have a cellular grid or a waffle, it's clipped inside an outer grid, and then you're producing the lamellas in situ. The transfer then goes via the vacuum capsule, hopefully, to the TEM to give you the transfer. And it's not automated in the sense that you're physically walking between the system, but of course you then have the well-aligned grid to pick up the workflow on the other end. In terms of selecting sites, we're typically using the fluorescence or maybe ion tile set overviews or electron overviews to give you some clues about the quality of your grid. And of course, you can see all of those channels presented to you as a user to be able to select which one gives you the information you need to choose the right sites. And so I think that's already helping a lot, I think, to choose the right areas for milling.
Great, thank you, John. Time for a couple more questions. One back to you, Mark. Does the Krios 5 come with a new generation Falcon camera?
No, the current Krios 5 is still comes still with the Falcon 4i. So all the capabilities also that Rijn talked to and that I talked to my presentation about this on-the-fly 8K motion correction, that's actually all basically leveraging more the Falcon 4i capabilities.
Okay. Great, thank you. And maybe the last question to Rijn. How can Tomo Live be parameter free?
It's a good question. We did a lot of work. Actually, our team did a lot of work to ensure that we do some good automation of which parameters to use. Making filtering automated in a way that you don't need to set up anything, but basically brute forcing the cross correlation. We're having detection of bad images. So in all the steps, motion correction, stack cleaning, the batch tracking, the pair alignment, the reconstruction itself, there are steps taken to based on the quality of the image. Look at, hey, what do I need to do here? That turns out pretty well, I have to say.
Okay, fantastic. And yeah, maybe just one more. How often do you have to calibrate the microscope? Mark, I guess that's one for you.
Yeah, and then you refer to the magnification calibration accuracy, which is improved from 5% on the, let's say, old generation Krios to 1%. As a user, this is something that service and factories is doing for you. The only exception that it has to be redone is basically when you decide to put a new camera on the system or when the column needs to be split after a service action. But those are really, I would say, rare occasions. So once the magnification accuracy set to 1% of the factory by service, you don't need to touch it anymore.
Fantastic, thank you. I know we still have some questions to go. I promise for anyone who's had a question that we haven't responded to during this session, we will reach out to you directly. But for the instance of time, I think we're going to call it a day at this point. Thank you so much, everyone, for joining. I hope you've learned a lot over the course of the last hour or so. Thank you to the panel and to Alex and Mark, and we hope to see you soon.